Hello everyone. Myself Dr. Neelam Mandal, Associate Professor from Botany Department from Government PG College, Panchkula. This video lecture has been prepared for BSc 6th Sem students of Botany. In this lecture, we will discuss the important topic of paper biochemistry and plant biotechnology. The topic is basics of enzymology. This is a broad topic. In the first part of this topic, we will discuss the discovery, nomenclature, and the classification of the enzymes. Let's start with the introduction. First of all, the term enzymology. Enzymology is the branch of science which deals with the study of composition and functions of enzymes. Then, what are the enzymes? Enzymes are biological catalysts and they are also known as the organic catalysts. Enzymes are synthesized by the living beings. And what is the meaning of catalyst? Catalyst is anything that speed up a chemical reaction that is occurring slowly. That means कोई भी रिएक्शन जो हो रहा है अगर उसमें हम कैटलिस्ट डाल देते हैं तो उसकी स्पीड इंक्रीज हो जाती है. तो एंजाइम्स भी यही काम करते हैं. एंजाइम भी रिएक्शन को स्पीड अप करते हैं. But these enzymes are synthesized by the living beings, so they are known as the organic catalyst and the biological or biocatalyst. They are also produced in plants and animals, so they are capable of increasing the rate of chemical reactions. So, enzymes are the biocatalyst that enhances the rate of biochemical reactions. About the nature of the enzymes, all enzymes are protein in nature except the enzymes which are RNA in nature. These type of enzymes are known as the riboenzymes. So the enzymes are protein substances and which are capable of catalyzing the biochemical reactions which are occurring in the living beings. An enzyme is a protein that enhances the rate of biochemical reactions but does not affect the nature of the final product. So the enzymes are capable of catalyzing the biochemical reaction which are taking place in the living being. Enzymes are effective in very small amounts. Very small amount of these enzymes are required to increase the rate of reaction. Or the enzymes are not altered or consumed during the reaction. That means they are remain unchanged at the end of the reaction. So the enzymes can be reusable. Again and again they can be used for the same type of reaction. Next is discovery of enzyme. Existence of the catalyst in the biological material was recognized in 1835 by the Jacob Berzelius in 1835. He also coined the term catalysis. He observed that potatoes have something which are responsible for the breakdown of the starch. In 19th century, in 1857, Louis Pasteur found that the yeast carried the fermentation in the living cells. Then in 1878, F. W. Kohne coined the term, and they first time used the word enzyme in place of the ferments. The word enzyme is formed from the two Greek words. En means inside, and the zyme, Zyme, which means the east. So the meaning of the enzyme is inside the east. Then is further in 1897, Edward Buchner was first to extract the enzyme from the yeast cell. This was the first cell-free preparation of the enzyme, and he gave name to this enzyme, the zymase. This zymase is cell-free preparation of the enzyme from the yeast. Then is the James Sumner in 1927 was first to extract the enzyme urease in pure crystalline form, and he also confirmed the protein nature of the enzyme. So after that the properties of the enzyme were studied in detail. Then about the nomenclature. Nomenclature means naming of enzymes. There are many methods for naming of enzyme. The old tribal names as pepsin and the trypsin. Some enzymes were named haphazardly and they not have any information. That is, they are not informative one, like the pepsin and the trypsin. Generally, these are known as the digestive enzymes. Then is the enzymes were named by adding the suffix ase, ase, to the name of the substrate. जिस substrate के नाम के पीछे ase लगा हो, that is known as the ase लगा हो, that is known as the enzyme. For example, lactase. Lactase, that means it acts on the substrate lactose. So the enzyme which act on the lactose, then the enzyme is known as the lactase. And the sucrase, that is acting on the substrate sucrose. So enzymes were named also according to the nature of the reaction they catalyzed. Like for example, oxidases, that means this oxidases name indicates that the type of reaction they catalyze is the oxidation reactions. And some enzyme have the two parts. Some of enzyme names have the two parts. First part indicates the type of substrate, and the second part, which ends with ase, indicates the type of reaction. For example, suppose this name of the enzyme is succinate dehydrogenase. That means the first part is succinate, indicate that the substrate on which it acts is the succinic acid, and type of reaction is the dehydrogenation takes place in this reaction. Then is this is the old system, then is the new system classification. The older system of classification and nomenclature has been modified by the Commission of Enzyme of International Union of Biochemistry in 1961. He classified this enzyme into 6 classes on the basis of type of reaction they catalyzed. The 6 classes are: first one is the oxidoreductases, then second one is the transferases, third one is the hydrolases, fourth one is the lyases, Fifth one is the isomerases. Sixth one is the ligases. Then the commission also have recommended the names of the enzyme by number. Each enzyme have their EC number. This EC number is consisting of 4 digits. What this digit indicates? For example, hexokinase is the name of the enzyme and their EC number is 2.7.1.1. What is this meaning? EC number means each enzyme has a numerical code which is formed of the 4 digits separated by the dots. First digit denotes the class, that is the class type of enzyme, reaction type of the enzyme. Second digit indicates, that means out of 6 classes किस क्लास को बिलोंग करता है वो एंजाइम. Second digit denotes the functional group upon which this enzyme acts. And third digit denotes the coenzyme. And the fourth digit denotes the substrate. For example, 2.7.1.1. What this indicates? This is the EC number of the enzyme and the first digit is the class. That means it belongs to the second class, that is the transferases. And the second digit indicates the subclass. That means in the second class, what is the subclass? That is the transfer of phosphate, that is the 7th subclass. And third digit indicates the subclass, that means alcohol is the phosphate acceptor. This is the first. Then is the last fourth digit indicates the specific name, that is the ATP hexose 6 phosphate transferase, which is known as the hexokinase. This is the reaction of that example, that is the hexokinase catalyzes the reaction, that is the reaction of the glycolysis, that is the glucose plus ATP then converted into the glucose 6 phosphate in the presence of enzyme. That enzyme is known as the hexokinase enzyme. Now let's discuss the 6 major classes of the enzyme one by one. The first class is the oxidoreductases. This class of enzyme catalyze the oxidation and reduction reaction. In these reactions, one compound is oxidized and the other is reduced. It means the substrate that is oxidized is regarded as the hydrogen or electron donor. That means there is the loss of electron takes place, and the compound that is reduced will be considered as hydrogen or the electron acceptor. That means there is the gain of electrons takes place. And these enzymes act on many chemical groups to add or remove hydrogen atoms or the electrons. The oxidoreductases are further divided into the subtypes like the oxidases, peroxidases, dehydrogenases and the reductases. Oxidases is the category of the enzyme that causes oxidation of substrate with the help of molecular oxygen. That means they transfer the hydrogen of its substrate to the molecular oxygen. And the peroxidases is the category of the enzyme. They cause oxidation of the substrate with the help of hydrogen peroxide means they transfer the hydrogen of its substrate to the hydrogen peroxide. Then these enzymes are known as the peroxidases. Example of the oxidases is the glucooxidase and the ascorbic acid oxidase, and peroxidases example is the catalase. And the other category of the enzymes is the dehydrogenases. These enzymes catalyze the oxidation of the substrate by removing of hydrogen from its substrate, but in this case the hydrogen acceptor is not the molecular oxygen. Hydrogen acceptor is the some other molecule. And the example of this dehydrogenases is the lactate dehydrogenase. And the other category is the reductases. These causes addition of hydrogen or an electron and remove the oxygen from the substrate. And these category of the enzymes are known as the reductases. That means it takes place the reduction reactions. Example of this reductases is the nitrate reductase, which helpful into the nitrate reduction reaction. In the next, the oxidation reductases example. These catalyze the oxidation and reduction reaction. In this reaction, you can see here is the hydrogen is removed during the oxidation of this substrate and it is taken up by the NADH. This is the coenzyme, and this type of reaction is the dehydrogenation, and the enzyme involved here is the lactate dehydrogenase. Clear? Then is the second class of the enzyme is the transferases. Transferases are the enzymes which bring about the transfer of the functional group or the chemical group from one molecule to the other molecule. The transfer group may be a methyl group, it may be aldehyde group, it may be a keto group, it may be any alkyl group, it may be amino group, it may be a sulfur group, it may be a phosphate. So the transferases are named after the name of the group transferred by them. कौन सा फंक्शनल ग्रुप ट्रांसफ़र हुआ है, मिथाइल हुआ है, फास्फेट हुआ है, सल्फर हुआ, उसके बेसिस पे ही ट्रांसफेरेजेस के नेम होंगे. फॉर एग्जाम्पल, इफ द अमीनो ग्रुप इज ट्रांसफर फ्रॉम वन मॉलिक्यूल टू द अदर मॉलिक्यूल, सो द कैटेगरी ऑफ द एंजाइम is known as the transaminases. If the transfer group is phosphate, then it is known as the phosphotransferases. If group is methyl group, then it is known as the transmethylases. So the name of the transferases is depend upon the name of the functional group. Example of this transferases, here in this reaction, alanine plus alpha ketoglutarate. Here is the amino group of the alanine is transferred to the alpha ketoglutarate. This amino group, here transfer group is the amino group, so the enzyme involved here is the transaminase. Alanine is the substrate from where this amino group is transferred. Then this converted into the pyruvate and the glutamate. Then is the third category of the enzyme, that is the hydrolases. Hydrolases the enzyme which bring about the hydrolysis of the various compounds. Hydrolysis, that means hydro means water, lysis means breakdown. With the addition of water across a bond, then the substrate is break down into the two smaller molecules. And this hydrolysis of various types, it may be esterases, it may be peptases, it may be glycosidases. It depends upon the substrate on which they act. If substrate is the esters, then it is known as the esterases. If the substrate is peptides, then it is known as the peptases. If it is glycosides, then it is known as the glycosides. Examples of this: if this category of enzyme is acted on the peptides of the proteins, then it is known as the protein hydrolytic enzyme. And if they act on the carbohydrates like the amylase, maltase, lactase, then they are known as the carbohydrases. They act on the lipids, then it is known as the lipase. Act on the phosphate, then it is known as the phosphatase enzyme. Example of this hydrolysis: these catalyze the hydrolysis reaction, where the water is the acceptor of the transfer group. Here in the fat, here is the transfer group is taken up by the water, then these triglycerides during this hydrolysis break down into the glycerol and the fatty acid. Here enzyme involved here is the esterases, because it is the category of the esters. Then is the fourth class of the enzyme is the lyases. Lyases is the category of the enzyme which cause removal of the group or direct breakdown of the substrate without involving any hydrolysis or the oxidation change. So that means the substrate is breakdown and with, but there is no hydrolysis takes place. At the place of the removal of the group, at that place the double bond is added. In the most of the cases they require the coenzyme, and the examples of this lyases is the fumarases, aldolases, carbonic anhydrases. The example reaction of this lyases is the from the glycolysis. You must be familiar with this reaction. The fructose 1,6 biphosphate. Here is this is the 6 carbon compound. Aldolases breaks this 6 carbon compound into 2 3 carbon compounds, dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. यहाँ से ये ब्रेक हुआ, तो जहाँ से ये ब्रेक हुआ वहीं पे ये इसका डबल बॉन्ड फॉर्म हो गया. So the enzyme involved here is the aldolase. Then is the fifth category of the enzymes, that is the isomerases. Isomerases is the group of enzyme which catalyze the isomeric changes within a single molecules. That means the isomerization takes place. That means internal rearrangement in the molecules of the compounds. and which carry out many kind of isomerization. It may be geometric one or it may be structural changes which convert a molecule from one isomer to the other isomer. Isomers are the molecules with identical molecular formula. Identical molecular formula means the same number of atom of each element, but there is the distinct arrangement of these atoms in a particular space. So, according to the type of this isomerism, they may be isomerases, it may be epimerases, it may be mutases. Example of this enzyme is in the isomerases phosphoglucoisomerase, and epimerases is epimerase and the phosphoglucomutase. So isomerases. Example is glucose 6 phosphate is converted into the fructose 6 phosphate. Isomerases causes the change in the position of the atom. Here is the position of the atom is changed. Then this aldehyde group like this glucose 6 phosphate, this is the aldose and it is converted into the ketose. Then is the enzyme involved here is the isomerase enzyme, and the name of the enzyme is glucofructose isomerase enzyme. Then is the second one is the epimerases enzyme that causes the structural change involving in the position of one carbon group. Xylulose phosphate is converted into the ribulose phosphate. Both are the 4 carbon compound, but their structural change is involving. One कार्बन एटम ग्रुप चेंज होता है बस उसमें, जिससे जाइलोलोज इज कन्वर्टेड इनटू राइबोलोज. दे बोथ आर द आइसोमर ऑफ each other. Enzyme involved here in this type of reaction is the epimerases. Then is the third category is the mutases. In the mutases which bring about a shift in a position of a side group within a molecule. साइड चेन की पोजीशन चेंज होगी बस इसमें. ग्लूकोज 6 फास्फेट 6 कार्बन कंपाउंड है, इसमें 6 कार्बन पे फास्फेट लगा हुआ है जो चेंज हो जाएगा ग्लूकोज 1 फास्फेट पे. 6 से फर्स्ट कार्बन कंपाउंड पे चला गया. देन इज दिस इज नोन एज अ म्यूटेशनल चेंजेस. Then the enzyme involved here is the mutases. Then is the last category of the enzyme is the ligases. Ligases are also known as the synthetases enzymes. These enzymes join the two molecules with the different or the covalent bonds. It may be CC bond, CN bond, CS bond, CO bond, like these bonds. And they catalyze the reaction in which two chemical groups are joined together, ligated, that means coupled together with the use of energy from the ATPs. Example is acetyl coenzyme synthetase. In this, acetic acid acetate combine with the coenzyme with the help of the ATPs, then it forms the acetyl coenzyme. With the help of the enzyme involved here is the acetyl coenzyme synthetase. Another example is the acetyl coenzyme carboxylase and the glutamine synthetase. Example here is catalyze the ligation, that is the joining of the two substrate. Here is the two substrates are glutamate and the amino group. Here they combine together, then they form the glutamine with the help of the energy required here, which is coming from this ATP. Here is the one amino group. This amino group is attached to this, then it is converted into the glutamine, and this is known as the synthesis of the one large substrate is formed. This is all about the all 6 classes of the enzyme, and these are the some questions related to these topics. Very short questions. Who coined the term enzyme for the first time? Why are the enzymes are called biocatalyst? Which enzyme is extracted for the first time? What are the ligases? There are some short questions are there. What are the function of the proteases? Differentiate between the enzyme and the inorganic catalyst, and differentiate between the dehydrogenases and the oxidases. How are the enzyme named? There are some long questions are there. Give one example of each reaction catalyzed by each of the following classes of the enzyme, like the hydrolases, isomerases, transferases. How have enzymes been classified recently? One long question. Discuss the IUB system for nomenclature of the enzyme. This is all about the discovery of enzyme, nomenclature of the enzyme, and the classification of the enzyme. In the next lecture, we will discuss about the chemical structure, different types of enzyme like the holoenzyme, isoenzyme, apoenzyme, and the different characteristics and the properties of the enzyme. Thank you.